Hey guys, welcome back. This time around we're going to begin our discussion of the reactions associated with catabolism. Before we do that, we need to appreciate something. And that something we need to appreciate is the initial substrate that we're going to be looking at each and every time. It's going to be a carbohydrate and how that carbohydrate goes through the process of metabolism. Now we'll be a little bit more specific and we'll say that the carbohydrate that we will choose is going to be glucose. Now glucose, if you recall, is also the sugar that we measure in blood. Now glucose will be transported to the cells via the blood and it will enter the cells using something known as facilitated diffusion. You recall what facilitated diffusion is. By definition, it is the movement of a molecule from a region of higher concentration to a region of lower concentration with the assistance of a transport molecule. And in this case, it would be a protein that's going to be found embedded in the cell membrane without the use of any metabolic energy in the form of ATP. Glucose will enter the cell using facilitated diffusion, and it will quickly be modified by adding a phosphate group to the end of it so that we can make it too large to fit back out through that same portal. This new molecule is called glucose 6-phosphate. Now that's going to have another advantage to us. It's going to ensure that the level of glucose inside the cell stays low at all times, because as soon as we add the phosphate group to it, it's no longer glucose. Now typically a cell does not have the enzyme necessary to do the reverse reaction, convert it back to glucose, with the exception of the intestines, kidney, and liver cells. As a result, we have glucose trapped. We have low concentrations of glucose then inside the cell by modifying it, which will permit us to continually facilitate diffusion to bring glucose in. Now let's take that glucose molecule that we have inside the cell and let's put it in these reactions. The first set of reactions that we have is glycolysis. Now glycolysis is going to be a set of 10 reactions that occur in the cytoplasm. Now these reactions can occur in the presence and or absence of oxygen. They do not require oxygen, so frequently we know these sets of reactions as the anaerobic respiration reactions. They are oxygen independent. We start with the glucose molecule as our initial substrate. The end product, if you notice from this set of reactions, is going to be pyruvic acid. In fact, it's going to be 2 pyruvic acids. So we start with a 1,6-carbon molecule, and it results in the production of 2 3-carbon molecules. So we've lost no carbon dioxide yet. But also notice, though, that we have lost one of our bonds. And that bond is a covalent bond between the 3rd and the 4th carbon. Notice through this process, though, as an overview, you'll see that the net production, though, will be of 2 ATPs and 2 NADHs. The NADHs are high-energy molecules. They'll be transported, as we've seen, to the electron transport chain. Now, we can also see that in the presence of oxygen here, the end product of glycolysis will go directly into the Krebs cycle. In the absence of oxygen, it will be converted to lactic acid. There are 3 phases associated with glycolysis. These phases are sugar activation, sugar cleavage, and sugar oxidation. Let's take a look at each of these 3 phases. Sugar activation. Sugar activation requires an input of 2 ATPs. We're basically priming the pump and starting the process. Those ATPs, we're going to use the tertiary phosphate group, and we're going to add first a phosphate group to the glucose molecule once it enters the cell to make glucose 6-phosphate. Then we're going to go through the process of converting glucose 6-phosphate to an isomer. Remember what an isomer is: same molecular weight, different conformation. We're going to convert it into an isomer fructose 6-phosphate, and then we're going to add the second phosphate group from the second ATP to that fructose 6-phosphate to make fructose 1,6-phosphate. This is a set of 3 reactions, as you see. These additions of phosphates require enzymes, and those enzymes that typically are adding phosphate groups to any kind of molecule are known as kinases. The conversion from a glucose to the fructose uses an enzyme in the family of enzymes known as isomerases. Seems appropriate since we are converting glucose to an isomer, fructose. Sugar cleavage is actually going to be the phase in which the covalent bond between the third and the fourth carbon of fructose 1,6-phosphate is cleaved, creating two 3-carbon molecules using an enzyme known as aldolase. This is the only covalent bond broken during glycolysis. This bond breakage will permit energy to drive the subsequent reactions in glycolysis. The end products of this cleave is going to be a glycerol 3-phosphate, which we know is G3P, and a dihydroxyacetone phosphate. Now that dihydroxyacetone phosphate is going to be quickly converted to G3P using, yet again, an isomerase. The third phase of glycolysis is sugar oxidation. Now in sugar oxidation, we're going to take the two G3P molecules and we're going to use the high energy that has been invested in them by the utilization of the two ATPs prior and the energy from the cleavage of the chemical bond, the covalent bond. We're going to take advantage of this high energy to realize in the initial reaction of the sugar oxidation will be added another phosphate group onto the G3P molecules to create 1,3-bisphosphoglycerate. The remaining 5 catabolic reactions will take advantage of this high energy state to transfer the phosphate groups onto 4 ADP molecules to generate 4 ATP molecules. Thus, we utilize kinase enzymes to generate 4 ATP molecules via substrate level phosphorylation. And we call this sugar oxidation. So where does the oxidation component factor into these reactions? In rheology, we will leverage that last bit of energy in these molecules to reduce 2 NAD molecules to produce 2 NADH molecules. These will be transported to the electron transport chain, where they will be utilized to generate ATP via oxidation phosphorylation. The end product will be 2 3-carbon pyruvic acid molecules. Thus, no carbons are lost as carbon dioxide during glycolysis. Now, if we were going to take a look at a summary then, we would see that during sugar activation, we use 2 ATPs to prime catabolism. In sugar oxidation, we generate 4 ATPs and 2 NADH molecules. The net ATP production in glycolysis is going to be 2 ATPs and 2 NADH molecules per glucose molecule. 1 covalent bond is cleaved, and the end product is 2 pyruvic acid molecules. Y'all have a great day. I'll see you soon.